few weeks myself, but I will say welcome back. And this is another one of our monthly programs to bring science to the Dutchess community. I will point out before I introduce tonight's speaker that on September 7th, our next program will be John Fitzpatrick from the Cornell Lab of Ornithology. Actually, he's director of the Cornell Lab of Ornithology. John has been a real shaker and mover in the ornithological and bird world, including his work to try to rediscover the ivory-billed woodpecker down there in Arkansas. But he will tell us about what's new in the world of bird ecology on September 7th. Tonight we have a special treat, namely one of our own scientists, Dave Strayer, who's been at the Institute for his entire career. Dave, like me, is a Midwesterner, and then went to Michigan State, where he graduated with high honors in zoology, then went on to Cornell and got his PhD in ecology, started here right after that, let's see, 1985, I think, something like that, and has worked his way up to the rank of senior scientist on the Cary staff. He's well known for a variety of studies in fresh waters, including studies of mussel and other mollusk ecology, looking at the invasion of the zebra mussel and what that has, both predicting what would happen and then studying what has happened with the zebra mussel in the Hudson River. Working with the team that examines the Hudson on behalf of the Cary Institute. He's been very involved with shoreline work and submerged aquatic vegetation, or SAV as we call it, and the importance of that for the ecology of the edges of the river and what that means for the reproduction of fishes and the general health of the entire ecosystem. And is well known for a variety of other studies on the Hudson. He has written a little book which will be available courtesy of Merrick Bookstore afterwards with Dave with a felt marker to sign your individual copy. The Iconic River, which is a fun little read about the Hudson, its history, what's going on with it now, and its future. And to tell us about the book here tonight, the man himself, Dave Strayer.
A few weeks ago, Bill and Lori Quellen, our PR person, asked me if I would like to talk with all of you about this book that came out a few months ago. I don't really want to talk about this book tonight. You don't really want to hear about this book tonight. But I thought I would use this occasion to talk a little bit about science and the future of the Hudson River. So I want to look in two directions tonight. I want to look backward at what scientific research has discovered about the river in the last few decades. And I want to try to look forward a little bit to think a little about the future of the river and the role science might play in the future of the river. So I want to look backward first at what scientific research has discovered in the last few years about the Hudson River. And I'm taking this book as the point of departure. This is Bob Boyle's wonderful book on the natural and unnatural history of the Hudson River. This was, the revised edition of this was published in 1979. It's still, I think, the first book that anybody who's interested in the Hudson, in the ecology of the Hudson River should read. It's a wonderful read written by a terrific writer. So I commend this to all of you. So what have we learned about the Hudson since 1979 when Boyle's most recent edition appeared? And I need to stop here and tell you that I'm going to be talking tonight about scientific research that's produced by all members of the scientific community. This is not my research. It's research by all these people and 100 more, including the research team here at the Cary Institute. I need to start maybe by reminding you about a little basic aquatic ecology. You probably know this stuff. This is what you learn if you read a textbook of freshwater or marine ecology. So the base of the food web are these little single-celled or multi-celled photosynthetic organisms called phytoplankton. Their growth is fueled by inputs of nutrients, chiefly nitrogen and phosphorus. This nitrogen and phosphorus can come in off the land as sewage, as runoff from lawn and farm fertilizers, as air pollution raining out of the air, the more nutrients you get running into the aquatic ecosystem, the more phytoplankton you get, sometimes to the point where the pond or lake gets undesirably scummy and green. And if you want to control that, you cut back on the nutrient inputs into the aquatic ecosystem. This phytoplankton also serves as a base of the food chain. The little animals that live in the water, the zooplankton, feed on phytoplankton, and they in turn are eaten by fish. The other important primary producer in this system are rooted plants, macrophytes living in the shallows. They produce food for the food web. They also produce dissolved oxygen, as part of photosynthesis. I think that probably this kind of thing is familiar to most of you. Almost nothing that I just said applies to the Hudson River. The Hudson River doesn't work like this, and I want to show you a couple of examples about what we discovered about how the Hudson River does work. So first, what does limit phytoplankton growth in the Hudson River? This graph here on the left shows the diagram of the water column at a fairly deep part of the Hudson River, like beneath the walkway over the Hudson. The water there is about 20 meters, about 60 or 65 feet deep. Tidal currents mix the river top to bottom. It doesn't stratify like a lake. And the Hudson, as you know, is not a very clear river, so it's dark. So this little line at the top shows light penetration as a percentage of full sunlight on like on a day like today. And you can see that almost none of the light penetrates down 5 meters. That's 15, 16 feet. By the time you get down here to halfway to the bottom, it's as dark as night. And down here at the bottom, it's darker than any place that most of you will ever be. This is a problem for phytoplankton because they're mixed all the way up and down and up and down. They get up here for a little bit, and they can photosynthesize a little bit, and then they're back down in the dark, and they're out of luck. It's like somebody strapped you to a conveyor belt, and you went past the cafeteria about every 3 days. So it's a hard place for phytoplankton to grow. The other thing we know about the Hudson is it's very rich in nutrients. So the two primary nutrients that limit growth in fresh water are nitrogen and phosphorus. The blue bars show the amount of nitrogen and phosphorus that came into the Hudson River before Europeans were here. These are low numbers. The red bars show the amount of nitrogen and phosphorus that are coming into the river today. The Hudson receives more nutrient inputs per surface area, than any other major estuary in the United States. It's full of nutrients. These nutrients come in from sewage, they come in from fertilizers, and they come in from atmospheric pollution, particularly in the case of nitrogen. They rain out of the air. There are ample nutrients for phytoplankton growth in the Hudson River. So what this means is that the phytoplankton in the Hudson River are limited by light, they're not limited by nutrients. And that statement has important management implications. There may be good reasons why you want to control inputs of nitrogen and phosphorus from the landscape running off into the water, but they're not going to change the primary production in the Hudson. We're not going to manage the amount of phytoplankton in the river, either up or down, by managing nutrient inputs to the river. So that's kind of an important thing to know. The second thing I want to talk about is this assertion that phytoplankton are the base of the food web. If you do a little budget for the Hudson River to see where the food comes from that might support the food web, this is what you get. Oh, I meant to say this. All the work, this tasteful little logo here in the lower right, this means that Cary Institute was wholly or partly responsible for the data on that slide. I'm trying to show you how important we are. So you can see there is some phytoplankton growth here, 12 grams carbon per square meter per year. There's a little bit of rooted plant growth in the river, a little attached algal growth in the river. But 90%, more than 90% of the food that comes into the river comes in from outside the river. It gets washed in from the tributaries that feed the river. It gets washed in ultimately from the landscape. This is tea and ground up leaves and things that are washed in from the landscape into the river. Now, if you're a phytoplankton proponent, you might object to this and you say, well, this is crummy stuff. This is delicious stuff. So the food, and that's true. The food web is running on this stuff, maybe, and this stuff, maybe this just all washes out to sea, and who cares what happens to it out in the sea, but it's not fueling the river's food web. But in fact, we have good evidence that a lot of the food web is running on this material that's coming in from the watershed. So first, If we look at gas dynamics in the river, it tells us that the river's food web consumes more food than it produces. Let me explain this before you look at all those dots. So we all know that photosynthesis consumes carbon dioxide and produces oxygen, right? Respiration does the opposite. It consumes oxygen and produces carbon dioxide. So if you had a system where it's producing more than it consumed, it would be producing a lot of oxygen. And the water would contain more oxygen than the atmosphere and less carbon dioxide than the atmosphere, right? This shows what happens actually in the Hudson River. This is oxygen in the bottom. This is carbon dioxide in the top. The data are measurements made, the black dots are measurements made in Hudson River water. The red line is the atmosphere. And you can see that the Hudson River contains way more carbon dioxide and quite a lot less oxygen than the atmosphere does. This tells us that the river as a whole is consuming more food than it is producing. And then there's another really neat study, and I'm sorry this is going to take a minute to explain, but this is just so cool. I have to share this with you. So we're all made of carbon and hydrogen and oxygen and all that kind of stuff. It's not puppy dog tails and what they told you. It's just elements. And those elements sometimes exist in multiple isotopes. So, for example, my body contains a lot of carbon. Most of it is carbon-12. A little bit of it, about 1%, I guess, is carbon-13. And a tiny little bit of it is carbon-14. If you study the isotopic composition of an animal and compare it to the isotopic composition of its possible foods, you can tell it's actually been eating. It's a sort of you are what you eat idea. And so what a group here at the Cary Institute did is analyze the isotopic composition of zooplankton. These are little animals that live in the water, and their possible food sources. And by doing that, and this is a tricky thing to do, but they did it anyway, by comparing the isotopic composition of the food and the consumer, they could determine what that animal is actually eating. Now here are the possible, so this is hydrogen-2, deuterium, carbon-13, carbon-14, doesn't really matter. The consumer should be close on this graph to its food. So here are the possible foods. Bill already introduced SAV. This is submerged aquatic vegetation. These are weed beds. This is floating leafed aquatic vegetation. This is phytoplankton. If this animal, if this zooplankton, were eating only stuff that was produced in the river, its isotopic composition would fall somewhere inside this triangle and this triangle. The other source of food is leaves that fall into the river from modern primary production. That's this dot up here. So if this animal were being fueled primarily by material that's produced in the river today, or material that was produced on the landscape today, the dot would be up here. The zooplankton dot would be up here. But what's going on? The dot isn't anywhere near that. The zooplankton is down here. It's very depleted in carbon-14. What's happening here is there's another source of potential food for the river's food web, and that is this aged terrestrial material. This is junk that the terrestrial ecosystem was not able to consume. So this is a tree that was photosynthesizing when Jesus was a little boy, and I mean that literally. And it captured carbon dioxide from the atmosphere in AD 3, and the leaf dropped to the ground. It was partially decomposed. The stuff that nothing ate was incorporated into the soil, and it sat there in the soils of the watershed for 2,000 years, untouched, junk. And then in the course of a few weeks, it flows into the Hudson River. It only takes a few weeks to make its way through the Hudson River. The Hudson River food web grabs that stuff and turns it into a zooplankton. So not only is the river's food web not fed by phytoplankton, it's fed by material, it's fed by photosynthesis that occurred on a fine summer day 2,000 years ago. And I think that's very cool. So we can cross this off too. The river's food web is not mainly or entirely supported by this phytoplankton as in this diagram. Okay, so what about plants producing oxygen? You all know that well-behaved plants produce oxygen. And here's an example of this. This is a well-behaved plant. This is wild celery. Those of you who boat on the river may know this is grass. There are grass beds in the river. There's lots of it. It's a submerged plant, lives in the water. And this graph, I'm sorry, it's a bit complicated. So this shows about 3 fine summer days, data from about 3 fine summer days, night, day, night, day, night, day. The blue line here is oxygen in the river, and it's dead flat almost. I told you there's not much phytoplankton production in the river, right? So the oxygen doesn't go up during day and night because there's not much going on as far as the phytoplankton is concerned. The green line here shows what's happening in these wild celery beds, and it behaves like a plant is supposed to. During the day, the oxygen goes up, and then during night it goes down as the plants respire, and they reach photosynthesis and respiration. Photosynthesis and respiration, everything's fine. Here's a plant that does something completely different. This is a horrible plant. This is water chestnut. It's an alien plant. It was brought here deliberately as an attractive ornamental from Europe in the 19th century. This is the green scum you see when you cross the Newburgh Beacon Bridge and you look down to the river, you see big beds of green scum on the east shore. This is what a water chestnut bed looks like. It's terrible stuff to work in. These guys, Nina and Heather, here probably took 3 days to get that far into the water chestnut bed. It has floating leaves, and the canopy of floating leaves is so dense that on this day, when they're sitting out here on this fine summer day, underneath this canopy of floating leaves in the water, it's darker than night. There's no photosynthesis occurring there underneath the water chestnut bed. So what happens is the tides move oxygenated water into this water chestnut bed. All the respiration from the underwater leaves, underwater stems of this plant, the roots of this plant, the bacteria and animals that live in the water underneath the plant, all that respiratory activity strips the oxygen from the water. So in just a few hours, the oxygen is depleted to 0. So this now shows the oxygen dynamic inside a water chestnut bed. And what you can see is the water comes in during the flood tide, nearly as well oxygenated as the river, and then it's rapidly depleted all the way down to 0. 0 is a problem if you're an animal, right? If we would all die in a 0 oxygen atmosphere, and a lot of fish would too. So this is kind of a problem. All right. So I crossed out the last thing on the simple diagram. The Hudson doesn't work like that either. That some of these plants actually have the effect of stripping dissolved oxygen from the water rather than producing it. So the reason I gave you these examples is that science has taught us how the Hudson River actually works, how it is actually put together. And that's not the way, necessarily, the textbooks say. This is a wiring diagram for a motorcycle, I think, that I got off the internet yesterday. And if you're a motorcycle repairman, you would want to have a correct wiring diagram, presumably. If you want to manage the Hudson River, it helps to have the right wiring diagram. And science is helping us get towards having the right wiring diagram for the river. The second bit of scientific progress is that science in the last 20 or 30 years has filled in a lot of details about how the Hudson works. These are things that we sort of knew in outline. We knew how the river worked, but we now have a lot more detail about how things work. And I'm going to give you 3 examples of this. The first is these plant beds. We know that beds of plants are important habitat for fish and wildlife. Those of you who are bird watchers or fishermen know that. Those are the places you go to look for birds or go to fish for fish. We also know that many of the plant beds in the river were destroyed when the river was converted to a navigation channel in the 19th and early 20th century. More than 4,000 acres of shallows were destroyed, either filled or dredged. So this is a habitat that we've lost a lot of. The state is concerned about it. We know there are beds of vegetation out there, but we don't really know how many there are, where they are, what they're doing, and how they're doing over time. So we've been working with a very nice group of people from the DEC, from Herner, from Cornell University, to map out where exactly these plant beds are and what they're doing. This is a beautiful, high-res aerial photo of the Hudson River. This is Kruger Island, for those of you who know that part of the river, Tivoli North Bay, Tivoli South Bay. And it shows this exquisite detail about where the plants are in the river. These are water chestnut beds that you and I can see very easily. If you look closer, this is a wild celery bed here. We have the most amazing photo interpreter working with us. I think she could see a gnat on a bee's back at 300 yards. And she could see all these little beds of aquatic vegetation out here in the river and turned this into this. This is a map of where all the weed beds, sorry, can't say that, all the beds of aquatic vegetation are in the river. And this survey has been repeated now, I think, 3 times, so we have some idea how the beds are changing in position. This information is very valuable if we're thinking about looking at like a permit application to put a new marina in along the river to see whether it's a sensitive area or not. Another area where we filled in a lot of details, what's on the river bottom. We have suspected for a long time that there is a bottom to the river, but we didn't really know much about it. And when I started working on the river in the late 80s and early 90s, the information we had were navigation charts. Navigation charts show water depths in feet here from lead line soundings, I think from the 1920s. And occasionally, these little cryptic notations indicating what kind of bottom it is. This is mud, and here's hard over here. Not very much. And this is important because many of the fish, and almost all the fish food in the river, lives on or near the bottom of the river. And it would be nice to know what's out there. So a group led by Lamont Doherty has done the most beautiful imaging of the river bottom all the way over the length of the Hudson River. And what they've produced are images like this. This is off of Kruger Island, so it's just north of the Kingston-Rhinecliff Bridge. This is the east bank and the west bank of the river to give you an idea of scale. The river bottom there is completely filled with moving sand dunes taller than I am. We had no idea. We just knew there was sand down there. I knew that because that's what you brought up when you sent a grab down. But it turns out there are these moving sand dunes here. This is the area around the Tappan Zee Bridge, which is now, we're going to replace the Tappan Zee Bridge, and it would be nice to have some idea what the river's like there. And what you can see is very detailed bathymetry. This is accurate to, I think, a centimeter vertically. You can see the fine details of the bed. You can see these 4,000-year-old fossil oyster beds that lie across the river, the direction of the river current there. And they've got this for the entire river now. The third detail example is we have a lot of real-time information on the river. When I started working on the river not that long ago, what you do is you go out in your boat and you fill a bottle full of water and you bring it back into the lab, and then either later that day or maybe later that week or maybe the next year, you'd analyze that bottle and you might find something interesting. You say, gee, on August 12th last year, something interesting must have been happening in the river. Let's go back and get another sample. And now we have real-time information where we can see what's going on in the river. If I were a little braver, I would go on the internet right now and show you what's happening right this minute on the river. We can do that. There's a group called HRICOS. I don't know what this stands for, Hudson River something. And they have a series of stations now along the course of the Hudson River and the Mohawk. And you can go online, anybody can freely go online and get 9 aquatic ecology variables and 12 weather variables at each of these stations. This shows what was going on at Norrie Point up till this morning when I downloaded this. And you can see that here's oxygen and temperature. And it looks like maybe there's an algal bloom out there. I don't know if that's true or not. But at least I can look at my desk and say, gee, I wonder if there's an algal bloom, and I can run down and get a sample now. There's still a lot of things we can't measure doing this, but we now have really good real-time information at a bunch of stations up and down the Hudson. So I would say we're kind of at the stage where we're moving in our understanding of the river from having sort of an artist's sketch of what the river looks like to having a more polished and complete scientific understanding of how the river works. All right, so I'm going to beat up this poor diagram just once more. The other thing that we've learned about the river is that it's very dynamic. It's changing a lot. In fact, if I had to name the single most important characteristic of the Hudson in the last few decades or even the last century is that it has been changing. So when you make a diagram like this, maybe we go out in 1972 and we say this is all true, and we measured it. And then we kind of assume it's true in 1982 and 1992 and 2002 and so forth. But the Hudson has been changing in important ways, many important ways, since Bob Boyle wrote his book in '79. I want to give you 2 examples. The first concerns non-native species, invasive species. This shows the time course of the arrival of non-native species in fresh waters in the Hudson River Basin from 1820 forward to the present time. We've brought in, either on purpose or accidentally, we've brought in more than 100 non-native species that are now established around here. we get a new species established in the area every year or two. It's about one species every 1.5 years. And this is a remarkably straight line, right? For ecology it is anyway. Which tells us that the rate of arrival of new species is not slowing down any. We're still getting a new species every year or two. In fact, in the last 5 years, here are some of the things that have turned up in the Hudson River Basin and established themselves. The Chinese mitten crab, the Asian clam, the northern snakehead, which we hope has been eradicated from Orange County, and the quagga mussel. So these species are coming in all the time into the river. Some of them have very large effects on the river's ecosystem. I'll show you this one example. This is zebra mussel. Bill already mentioned this. This is an animal that we've worked on a lot here at the Cary Institute. So these are zebra mussels. They're native to Europe. They were brought in accidentally in ballast water into the Great Lakes in about 1985. They turned up in the Hudson in 1991. Since 1991, they've been the most abundant animal in the river. In fact, if you were to take a balance and put all of the native animals on one pan of the balance, all the fish, all the shellfish, all the insects, all the worms, even all the bacteria on the one pan of the balance, you put just zebra mussels in the other pan of the balance, zebra mussels outweigh everybody else in the ecosystem combined. So this is kind of a big deal. A good way to look at how many zebra mussels there are in the river is to look at how much water they filter. These are filter feeders, so they make their living pumping Hudson River water through their gills and pulling out the edible particles. So this is the amount of the river water, of the water in the river that they filter every day during the summertime. And so it's somewhere between, say, 20 and 100% per day since they arrived. This little dashed line here is the combined filtration activity of all other filter feeders in the river. So starting about 1992, the structure of the Hudson River ecosystem changed dramatically, and nearly everything that we measure about the ecosystem also changed dramatically. This shows the changes that we observed in about the first 10 years of the zebra mussel invasion, and I'm not going to go into this in detail, but you can see generally a withering away of the planktonic part of the food web. Phytoplankton went down by 80%, and so anything that was hooked to phytoplankton, anything that depended on phytoplankton also declined. Because the river got a little clearer here, then we also had a flourishing of the food web in the shallow water parts of the Hudson River ecosystem. My point here is just that after the zebra mussel arrived, the Hudson River ecosystem was a fundamentally different system than it was before the zebra mussel arrived. Different wiring diagram. The other thing that's very interesting that we're observing with the zebra mussel invasion is that the river hasn't settled down to any kind of equilibrium yet. And again, I'm not going to show you a lot of detail, but a couple of things here. This shows the annual survivorship of zebra mussels from 1990 forward in the Hudson River, in nature, in the river. This is the probability that a zebra mussel is 1 year old will live to blow out the candles on the birthday cake when it's 2 years old. And you can see that they used to have a pretty good chance of that, about 50% chance. Now it's less than 1% of them live out that year. So something is killing the zebra mussels young now. We think that this is partly blue crabs and it's partly something else. But the point that I'm making here is just that things have changed a lot, and we've seen a recovery now of some parts of the ecosystem. So here's the small zooplankton. There used to be a lot of them in the river. Zebra mussels came in, they ate them all. Here's they ate them all phase here. And then in the last few years, we've seen a recovery of these of small zooplankton. In fact, we've seen a recovery of many of the kinds of fish food in the river, which is an encouraging sign. So the zebra mussel impacts are still changing. The story's not finished yet. The river's still a moving target. The other story about change I want to tell you is a really beautiful bit of scientific work that Ike Wergen from New York University has done on the tomcod. This is a tomcod. They're little codfish, less than a foot long, and they live in the lower parts of the Hudson River estuary, maybe from the Newburgh Beacon Bridge south. Because of the pollution in the river, the tomcods have been subject to enormous rates of cancer. This is really, I'm not showing any pictures, this is really horrible. So more than 90% of the 2-year-old tomcod in the Hudson have either liver cancer or precancerous lesions. If you go elsewhere where tomcod live in clean rivers, they don't have this. So this is clearly a response to the pollution in the Hudson River. Now, the really interesting thing that Ike's team has found is that, well, I should back up. So they think this is a result of the PCB contamination from the General Electric plant, right? And also an herbicide factory down in New Jersey that released a lot of bad things into the river between 1940 and, say, the 70s in both cases. Since that time, these fish have evolved to become 100-fold less sensitive to these pollutants than tomcod elsewhere, just in a few decades. People think about evolution being, you know, the relentless march of time and Tyrannosaurus rex and millions of years and so forth. This has happened since 1940, probably since 1970. This shows the frequency of 2 genes in tomcod in a bunch of clean rivers here, and then 2 dirty parts of the Hudson River estuary, the Hackensack and the Hudson River. The black is probably the native genotype. It's very sensitive to pollution. The white is the insensitive genotype. And you can see that in the polluted areas, this insensitive genotype has already come to predominate just in a few decades. So evolution has been occurring in the river in response to the changed conditions in the river. All right, so let me try to briefly summarize scientific progress since Bob Boyle's book in 1979. There are 3 areas where we've made, I think, really important progress. We have better conceptual models of the river, better wiring diagrams. We have much more detailed information about things in the river, and we have a better documentation and understanding of change. Now, if we had about 15 more hours, I could go through all of the examples of this. I just cherry-picked a few examples of this tonight, but I don't want to give you the impression that what I talked about so far is all scientists have learned. Scientists have learned much more than what I've said tonight about the river. And that's basically why I wrote this book. I think it's just really interesting what people have found out about how the Hudson River works. Most of this information now is readily accessible to anyone who has access to technical journals and has a PhD so they can read the darn things. And I thought that a bunch of other people might be interested in this. So this little book is an attempt to kind of summarize what scientists know about the river in English. And I don't know if it works. I want to make a little digression here. Is this scientific progress I've talked about on the Hudson, is this happening all across the United States, or is there something special about the Hudson? There's something special about the Hudson. So this shows the amount of scientific information that has been published on a bunch of American rivers. These are all rivers that are about the same size as the Hudson River in terms of the amount of water they carry. And on the y-axis, so I don't know, here's the Connecticut, the Delaware, the Susquehanna. These are nearby rivers, and the other ones are just an assortment of rivers I just grabbed. On the y-axis is the percentage of the scientific attention on the Hudson that has been focused on these rivers. And you'll notice that the high here is about 20%. So the Hudson is about 5 times more well-studied than the next best studied river in this set. And some of these rivers, like the Washita, which is a perfectly nice, interesting, and important river, has received about 1/50th of the study of the Hudson. So the Hudson is not typical. The Hudson is extremely well-studied. It's extremely well understood compared to any other river in the United States and indeed in the world. Why is this? And I want to bring a few factors responsible for the scientific scrutiny to your attention tonight. The first is the Hudson River Foundation. Some of you probably know about the Storm King settlement, where there was a big, I guess about a 20-year legal argument about building a pump storage plant down Storm King Mountain in the Hudson Highlands, and about water withdrawals for other power plants in the Hudson. One of the elements of that settlement that was finally reached that is probably not well appreciated was they set up a scientific foundation, the Hudson River Foundation. The power companies kicked in, I think, $12 million to start it. And the purpose of this was to study the Hudson River. The idea was that maybe if we studied the river better, we wouldn't be tied up in court for 20 years arguing about how the river worked and whether the power plants were filling straight gas or not. So the Hudson River Foundation has been really instrumental in funding work on the river, in serving as sort of an honest broker and getting scientific work done on the river. We also have an extraordinary program here at the New York State DEC, the estuary program in particular, run by Fran Dunwell here. And they funded some work on the river, research on the river. I think more importantly, they encouraged just a really vigorous, open two-way dialogue, or multi-way dialogue, I guess, between scientists, between managers, between various stakeholder groups on the river. People routinely, and this is going to sound really stupid, I suppose, but we routinely talk and email to each other about what is important, what we're finding, what impediments we have. And this is not typical of all government agencies in this country. And we're really lucky to have this one. And there have also been there are a number of education and advocacy groups. And I'm going to get in trouble by showing these three logos. These are three really important groups that have been very successful, but they're not the only groups. There are other groups that have also helped out here. But these groups have made local people aware of the Hudson River. How many of you have been out on Clearwater? Lots, right? Yeah. And they made people care about the river. They made people pay attention to news about the river. They've made people write letters to their legislators about the river. They've made people feel proud of the river. Many of you are probably here tonight, ultimately, because one of these groups engaged you in Hudson River stuff. The other thing these groups do that people don't know as much is they sue the bastards. And so sometimes it takes legal action to move things forward on the Hudson River. And these groups and other ones have been in court to look out for the interests of the people of New York and the interests of the Hudson River. So the Hudson is not typical in terms of the amount of scientific research that's been done on it. And it's, and us too. We've had a really good group of scientists here. I'm talking about my colleagues now, not me, who've been working on the river for 25 years. These are some of the brightest aquatic ecologists in the world, literally. And we have been working on the river for 25 years. We've engaged a group of more than 100 collaborators and students from other institutions to work with us. And so we've had this remarkable coming together of a number of groups and a number of different interests that have made the Hudson so well studied. Okay. So, I don't know how many of you have written books, but there's some point where you finish the book, you finish the darn thing, you submit the manuscript maybe, or maybe you make the last correction to the page proofs, and you send it back, and there's this wonderful feeling of satisfaction, of elation even, lasts for about 4 minutes. And then you start thinking about all the stuff you did wrong. All the stuff you left out, all the figures you didn't check, all the awkward phrasing, all the inept analogies you used. And I'm not going to go through all of my personal list that my brain helpfully brings to my attention every night at 3 in the morning. But there's one thing that's so obviously missing from this book that I want to spend the rest of the talk on that subject, and that is the Hudson's future. How come there's no chapter in the darn book about the Hudson's future? In a way, that's the only interesting thing to talk about in the Hudson is its future. So I actually felt bad enough about this or something that I had drafted a 13th chapter to the book. And any of you who buy the book, Allison has copies of this unpublished Hudson's future chapter, and you can tell me whether you think it's the right thing to do or not, but I did it. So I want to talk a little about the Hudson's future now. And you, hold hands now. So you understand this is an uncertain endeavor, and I can't cover all the things that are interesting to think about with respect to the Hudson future. So I want to just raise just a few points about the future of the Hudson. I said a little while ago the most characteristic thing about the river in the 20th and early 21st centuries has been change. And we can expect the river is going to change tremendously in the coming decades. I want to talk briefly about 3 kinds of change that I think that we can expect. They're almost certain to occur in the river. The first is it's going to change in response to deliberate management. This is a happy story. We're now, we have very stringent regulations on the harvest of fish like the 2 sturgeon species, the American shad, the river herring. Some of you have been following these stories. It's sad in a way that we've had to shut down traditional and long long-established fisheries for these fish, but we are working together with other states up and down the East Coast to bring these fish populations back to where they were in the past. We're also trying to fix some of the toxic pollution problems in the river, and the most obvious one is the PCB dredging that's going on right now in the upper river. But we've also stopped inputs of a lot of toxics as a result of the Clean Water Act in the 70s. So the river is getting better as a result of that. So we can expect, in response to this deliberate management, to see a better river in some respects in the future. So just to people, I say I'm too depressing when I talk about the river, but I'm going to give you a couple of happy stories. This is what happened when we started doing effective interstate fisheries management for striped bass up and down the East Coast. They were at historic lows in the 1970s. We imposed regulations on harvest all up and down the East Coast. This shows the number of baby striped bass in the Hudson River from 1975 forward. This is a logarithmic scale, so every tick here is a 10-fold increase. And we've come up to a point where there are something like 100 times as many baby striped bass in the river as there were in the 1970s. Fisheries management can work. Habitat restoration can work as well. The PCB contamination is not the only Superfund site on the Hudson River. The other one is Foundry Cove near Cold Spring. This was the site of the Marathon Battery Factory. They dumped huge amounts of cadmium and nickel into this wetland. It was remediated a few years ago. They cleaned out all the contaminated sediment, recapped it, replanted it. This is Foundry Cove today. And more importantly, these are the cadmium concentrations in Foundry Cove before the restoration project, and then in a series of years after the restoration project. So things can get better if we go out there to make them better. The second class of change is a little less happy, in my opinion. These are things that we can see they're going to happen. We're not managing them very well right now. So these are things that we don't really understand entirely what's going to happen. We're going to have to live with the consequences. We're not in a position where we're going to be able to prevent them. The two big examples are climate change and the arrival of more non-native species. These are regional to global issues that can't be solved entirely by local action. And we haven't solved them, right? Climate change is going to happen. More species are going to move around. And here are the sorts of things we're going to be looking at. This is temperature at New York City projected under a number of different scenarios. This is sea level rise at New York City, a number of different scenarios. You've all heard this before. We expect the temperatures to rise substantially in the coming decades. Sea level rise has been hard to predict, but the best estimates are 2 to 4 feet by, see if I get this right, 2080. And the Hudson is at sea level essentially all the way to Troy. So we're going to see the water level rise all the way to Troy. There's also been some suggestions. We're going to see more extreme events. We're going to see more severe storms than we used to. So the river's going to be warmer. Just the mere fact of the river being warmer is going to have 1,000 hard-to-predict ecological consequences. The same with sea level rise. It'll change everything about the ecology of the river. The other big unknown here is human responses to changing climate. We're not going to just sit and let this stuff happen. There's already discussions about installing surge barriers in New York City, for example, on the Hudson River, multi-billion-dollar project. There's already talk about building up shorelines to prevent flooding and flood damage along the shorelines. These human responses may cause great ecological damage, especially if, as we usually do, we wait for a crisis to act and then build something that's not well considered. The ecological responses to all this almost certainly are going to be large. They're going to be pervasive. They're going to affect everything in the river. And I don't know any ecologist who'd be willing to make a firm prediction about exactly what the Hudson River is going to look like as a result of these changes. We're going to see more of this kind of thing. This is last fall's. These are pictures taken during last fall's floods. And you can obviously see the challenge to infrastructure, right? This is the Metro North line at Croton Harmon. This is the Newburgh waterfront. People are almost certainly going to want to do something, build something protect this infrastructure. The other thing that you can think about when you look at this picture is if there were vegetation beds here, if there were a wetland here, it would be drowned out. It's going to be drowned out by more water. And it's unlikely that the river will build up its bed fast enough to allow those shallow water habitats to remain in place. I already showed you this graph about biological invasions. Almost certainly we're going to continue to see new invaders until we start doing something in this country effective about preventing biological invasions. So we're now holding our breath to see what the mitten crab is going to do in the Hudson River. We're holding our breath to see if we can do something about the bighead carps and keeping them out of the Great Lakes. If they get into the Great Lakes, they will move into the Hudson River next. And these are hard to predict exactly what's going to happen. We don't know which species are going to arrive. We don't know when they're going to arrive exactly. We don't know exactly what they're going to do. But I think we can count on there being large impacts. And the third class, which everybody forgets, are surprises. It seems like when planning forward, we always think, well, we're going to be able to plan for everything. We're smart. We're going to think of everything. But if you think about a lot of major changes that happened in the Hudson River, the PCB contamination of the river was a surprise. The sea level rise, Bob Boyle didn't write about sea level rise because it was unanticipated when he was writing his book. He didn't write about zebra mussels because it would have been a surprise. And so I think we can count on, we better count on, having surprises in the future. Things are going to happen that we didn't anticipate. Things are going to happen that we don't understand when they happen. So here are a few things to think about in terms of how we might respond to what I said. The Hudson is going to change. The Hudson is going to change. What this means is that even if we have perfect scientific knowledge of how the Hudson River works in 2012, and even if we have the perfect management scheme to manage the river in 2012, if we just stop and put those things on the shelf, so that's what we're going to do for the next 30 years, those are going to be bad in 2034. The river's going to be a different river. The wiring diagram is going to be different. So the scientific understanding and the management plans are going to be ill-fitted to the new changed river. That suggests to me that we're going to need to continue to study the river. Scientists are not going to be able to just go off and say, well, we did that. The book, some Strayer wrote this book. I guess we're done. We're going to have to continue monitoring the river. Monitoring programs are always on a chopping block. It seems like that they're unsexy, and people always say, well, they cost us money, let's cut those monitoring programs. That's a bad idea. This is going to sound really stupid too. We need to use scientific information to manage the river. This is something we're doing a really good job on here with the estuary program in New York on the Hudson River. We should not assume this is just going to keep going on in the future. You look at North Carolina. You guys been following this debate in North Carolina where they're arguing about whether planners are allowed to consider sea level rise in planning for the future, or whether they're legally forbidden to consider sea level rise. So people don't always use scientific information, even if it's there. We should also be prepared to shift course. The river's going to change. We're going to be surprised. And this challenge of developing flexible management structures is something that I think we probably need to work on. And then finally, the people in the Hudson have dealt really well with local issues. They haven't dealt really well with regional to global issues, things like climate change, things like species invasions. These are the hard problems for the future. And I think that we're going to have to struggle a little bit to get locally interested people engaged in regional to global issues. We're going to have to think locally and act globally for a change. All right. 2 closing comments, and then we can all go out and eat some more cookies. So I want to consider briefly what role science plays in environmental management and whether science is keeping up with the demand for environmental information. That is, are we going to have the science we're going to need to meet the management challenges of the future? So sometimes I hear scientists say, and members of the public say, that what is really needed in environmental management is better education and advocacy. And I don't want to be misunderstood here. Education and advocacy is absolutely essential to wise environmental management. And we only need to look at the example here in the Hudson to see this. Groups like Clearwater and Scenic Hudson have been immensely influential in improving conditions on the Hudson River. And the management agency. I mean, where would we, you have to have somebody there who's managing the river. But it seems to me you also have to have science that's equally important. What do you educate people about? Would it have been helpful for us to educate people about that textbook aquatic ecology diagram and tell them that's how the Hudson River works? Would it have been helpful for a management agency to manage the river as if that textbook diagram were how the river really worked? So it seems to me that science occupies an essential place at the table, just the same as the management agencies, the education groups, the advocacy groups. All of them are essential. Arguing about which one is more important than the other is like arguing whether your kidney is more important than your liver. Now, I have some bad news, I think, about whether we're going to have the science we need to support wise environmental management in the future. I'm concerned by this right now. This is a very nice diagram showing the amount of money that's been spent on all kinds of Hudson River research over the last 20 years or so. It came from Dennis Zakowski at the Hudson River Foundation. The Hudson River Foundation is in blue here. They were the big funder through the 80s and 90s. And you see that they are not funding as much research as they used to. In fact, overall, the amount of research money being spent on the Hudson River has gone way down from the peak. Have the problems of the Hudson River become less severe all of a sudden? Or do we have less challenges? Have we solved all the problems that we need to solve on the Hudson River? I don't think we have. And so this concerns me. We see the same thing on the national level. This is the data from the National Science Foundation, from the core programs that the Cary Institute gets most of our funding from. And I want you to focus on the green line. This is the success rate. This is the percentage of proposals that are submitted that are actually funded. I'm suspicious of these data. They're way too high based on, I'm serious, based on the data that we've seen. But I think the trend is correct. Success has fallen by about half in the last 10 years. And I don't see any signs that this is likely to increase in the near future. So I'm quite concerned about whether we will have the science in 10 years, in 20 years, in 50 years that we need to confront these environmental challenges with. All right, so the future of the Hudson. The future of the Hudson is pretty wide open right now. And I think probably the most useful thing I can say about it is that we can be pretty sure that we're going to be faced with some serious challenges in managing the Hudson in the coming decades. And the river we leave to our grandchildren, to future generations, is going to depend on how well we deal with those challenges. Thanks.
David graciously volunteered to take a few questions, and then we'll get on with some book signing. But right here.
Why do you think the Hudson has been so much more extensively studied than other rivers? How did that happen?
Well, I think it's partly back to people who have lived on the Hudson and who care about the Hudson. So if you trace the Hudson River Foundation back to the Storm King settlement, that came out of scenic Hudson, I think, largely. And scenic Hudson came out of people who loved the river, who cared about the river, who weren't willing to view the river as the property of the Consolidated Edison Company, ought to belong to the people of New York. Other rivers haven't had that, and I think there's a positive reinforcement there once that gets going. Other rivers haven't had that. I grew up in Michigan, and we accepted that the steel company owned the river. And when they polluted the river and got fined $50 for killing thousands of fish, we were just like, it's just the way the world works. And people here didn't accept that. I think that's why. Yeah.
Can you speculate on potential impact of hydrofracking if it goes forward in New York State on the rivers including the Hudson? And is there any data on that in other states?
There's a little bit of data, not very much. And this is a perfect example of where it would be nice to have more science to support the environmental decision making. I don't know yet. I don't think anybody knows yet how hydrofracking is going to affect New York's rivers or the Hudson. Because we don't know yet how we're going to deal with this issue in New York. at least we're not dealing with the way that they've dealt with it in Pennsylvania, which is to go ahead and permit it before they understood the impacts. And I'm hopeful that that we will consider the environmental impacts well in New York before we actually start hydrofracking. I'm hopeful about that, I guess.
Here.
You mentioned General Electric. General, what should they do when they have a spill like that? Should they dredge or not dredge? What do you think about that?
I supported the dredging project. It was I thought, the better of two bad choices. The good choice, we had two good choices. First good choice would have been not to dump the stuff in the river. And it wasn't, didn't need a permit to do it, but they knew it was a human health hazard at the time they did it. So, and the second good choice would have been not to take out Fort Edward Dam, because it was all piled up behind the Fort Edward Dam until 1973. And PCB contamination was discovered, I think, in 1970 in the river, and we didn't act fast and smart enough to leave that dam in place. And so that left us with two poor choices, which were to dredge at large cost to partly solve the problem, or to not dredge at all and be faced with a crippling problem for a long time.
One more, maybe? How about right here in the center?
How do we encourage more money to be funded for scientific research?
You know, I meant to say that. So I doubt that many of you are in a position to write us a check. If you are, Bill would be delighted to talk with you afterwards if you want to support some scientific research. You can, all of us can write letters, though, to our congresspeople. I understand that there are issues with the federal budget right now. NSF, the National Science Foundation, not every president, many presidents have promised more money to research. Everybody likes to say that scientific research is good. There's these plans keep coming up for doubling the NSF budget, and then they keep getting cut back. And I think one of the reasons they keep getting cut back is that we're not, and I'm just as guilty as anybody, we're not spending enough time screaming at our congresspeople about this.
Dave, many thanks. It was a wonderful review of your work, the river, future of the river, and just a sobering look at what faces the science in this country. For those of you that do have your checkbook with you, I'd certainly welcome you to join our Aldo Leopold Society, which guarantees you a seat here at the Friday night lectures. You can see there's a few people that had them reserved that didn't show up, but that allows me to point out those name tags. And it is a way to ensure that you get seating at our Friday night lectures, as well as a number of other privileges of supporting the Cary Institute. But let me at this point thank you for coming. Point out September 7th, John Fitzpatrick of the Cornell Lab of Ornithology will be our speaker. And the Friday night at Cary series will continue on through the coming year. And I welcome all of you to come and join us. And right now, let's go join Dave for that promised cookie while they last, and a chance to buy his book.